السلام عليكم ورحمة الله وبركاته. أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقهوا قولي. Inshallah ta'ala, we'll be getting started shortly. I'm just going to be taking an attendance, but inshallah ta'ala, we'll be focusing on the viable counts and chemical germicides with some tests on plates within ta'ala today for different bacteria. May Allah subhanahu wa ta'ala say. Okay, everyone. So we're gonna go straight ahead. I know some of you are counting, which is great. Your Bible counts. When your group is ready, they can draw it, or I'll let you know what we need to do. First off, uh, okay, everyone. So likely many of you are doing the Bible counts. That's amazing. It's a great way to prep ourselves for the generation time assignment, which we'll talk about later on today. But folks, I want to give some important updates, important guidance from the notebook quality, the lab performance, and updates that I've talked about on Focus. Hopefully everyone has seen those details, but I'll briefly touch upon a few important points. Everyone's paying attention, by the way? It's okay if someone in your group is counting, but please have open ears accordingly. Um, so as I touched upon, some of you are doing the viable counts right now. For your plaque assay, I'm going to talk about that because considering you have 2 more days, for those that haven't started, I would just give you some kind advice based on what I've talked about before. Hopefully from last week you recall, but essentially you need to have detailed information from the intro, going from generic from the lytic cycle, for example, all the way down to your research question with specific details noting the bacteriophage, the bacteria of interest, and talking about the significance of it there beforehand. That's your intro. Methods is not required. Results, you need to have figures. Once again, I'm going to highlight this in relation to your notebooks. Some of you are missing exercises. Some of you are missing details in your captions with specific annotations for the letters of each panel or each plate that you're using. Please bear in mind that as notebook quality is important, the same applies for your FLR, and it is representative, i.e. if you are not putting as much effort as you should be for the lab notebook and its quality and completion, this not only reflects that component of your grade, but also importantly, like for the lab notebook, but also for your FLR. So bear that in mind, folks. I want you to do your best, but you have to also put in the effort. Now, other points in terms of anomalies, some of you from, let's say, plate A, your control, may see only colonies, small colonies, or maybe you don't see anything at all. That's something that you definitely want to talk about. Talk about the anomalies from your methods in your discussion. Also, for your results, you need to be able to provide the PFUs for your plates from all of the ones that you're able to count. And if you weren't able to observe, let's say, for example, too few to count for the number of colonies in a specific plate, or there were too many beyond the range of 30 to 300, then you essentially have to include that as your observations, either in a table or in Word format. Any questions regarding that? Briefly as well, your discussion includes summary of your objective, the results, with your mean, by the way, your mean PFU, i.e. as you collect all your 4 plates together. Remember, even if there's only 2 out of the 4, but ideally 4 plates that you're able to count within range. You need to give me your mean calculated, essentially plaque density, i.e., that way you'll be able to derive the original density of your bacteriophage, original population density. Yes. No. So when it comes to, for example, let's say you have 2 plates. One is, like again, 242 colonies, 10 to the power of 6, a -6, essentially. For context, we say divided by, but in a nutshell, if you divide this by a negative exponent, it's like multiplying it by a positive. So essentially, this is 2.42 times 10 to the power of 8 PFU, right? As an example. Now, another example is, let's say I have 31 times 10 to the power of 7. You're going to get 3.1, right? We could say yeah, 3.1 times 10 to the power of 8.
And then you add them together and that divide by 2, and that's your mean?
That's correct, yes. Okay. That's what you would do.
And that's what you're getting the space.
Yes, that's what you get, and then you would mention that not only in your results, but also in your discussion. So bear that in mind, folks. In your discussion, include the extensions, limitations, the summary, as I mentioned before. If your results are as expected for the original cell density or original population density of your bacteriophage. Also, make sure that your title is specific, or at least that you provide the lambda bacteriophage in your annotation. Remember, it's not simply any bacteriophage, it's the lambda bacteriophage. And you could see further details in your methods from the lab manual. Any questions about the plaque assay formal lab? That's correct, but you need to provide for me your observations for the other plates, i.e. if there were too few to count, ideally you could tell me that number, but if you can't, that's okay. If there are too many to count, also mention that. You should also mention it for your control, i.e. if you see a large smear, if you find single colonies, or if you don't see anything at all, you want to provide your observations accordingly for all results that you've obtained. Does that answer your question, Aleen? Yeah? It can actually. So there's a great question. By the way, am I pronouncing your name correctly? Connor. Okay, so to clarify, folks, I mentioned this before. There's 3 different limitations generally. One is the accurate volume that you used, right, for the bacteriophage, for example. If you use too much, or too little, then that would, of course, influence or potentially influence your results. I forgot to mention this as well, but before the end of today's lab, please send me either via email, please send me your plaque and viable count data accordingly as your groups, so that way I can keep track and if needed, look over it in due time. Now, the other 2 limitations include aseptic technique, right? Because if you don't see anything, let's say in your control, you may have already heated up your L rod too quickly or added in 90% ethanol and essentially sterilized your bacterium without letting them to grow. So that's another one. And yeah, cross-contamination is another, right? So if you perhaps see different sized bacterium, ones that are obscurely larger than the ones that you would have seen from E. coli, then that's also a hinting tell of you perhaps seeing anomalies. That being said, like if you see others and if you're aware of others based on the methods, then you can describe them accordingly. It also resolves around other components, units as well for timing that you may have used that could be of variability from your results. So you can just bear those different points in mind. Any other questions? Everyone, by the way, has been able to count down their viable counts for the generation time? Yes or no? Not yet? Viable counts. Has every group done it at least? Or is in the midst of doing it? Yeah? Okay. In the meantime, let me just go over the slide. Okay, so okay, so hopefully everyone can see the slides beside me. Okay, let me just start off with a few important things. Unlike what you see, or something as an extension of today's class, just pay attention, folks. For exercise one, I'm going to be doing equalization of your ODs for Staphylococcus epidermidis and E. coli. You all should have the tubes that you need. However, there are additional tubes here. You have obviously water here for those that are essentially collecting the germicides. Have them also here. We'll be sharing them as benches. So each set of groups will be sharing a specific germicide. To bear that in mind, you may have to oscillate accordingly. But you have as well the other materials that you need for today. The catalase test for hydrogen peroxide is in a separate set of tubes, these smaller ones, whereas for the germicide, you'll find them in the larger conical tubes shown here. Okay. We have the liquid broth that we'll eat shortly as well. I just want to highlight as well. You have pipettes available on that side with tips on the other side. So to be able to collect any additional materials that you need for today on either end, just make sure that you return them back to their appropriate locations, either here, down there, or on the other side. Yes. But if you guys want to split up, that's also okay because the last two exercises 3 and 4 are generally easier to be done separately. So as pairs, that's also okay within your groups of 4 or 3 to 4 accordingly. Any questions, at least for materials and what you guys need? Let me just go quickly then. Oh yes, perfect. So for those that were wondering about like the results as well for post lab for lab 5, you can have a look there. Let me see. So I talked about a few things. Let me just also clarify for updates to that. Staphylococcus epidermidis and E. coli are grown even though at 24 hours roughly. They have different turbidities. As you can actually see if I look the plot up. This is Staphylococcus epidermidis, right? I have to keep in mind resuspension. And this is actually E. coli, right? So you can see quite a difference between the two. And so this is actually important because technically, when you do your results, and I'll share with you guys which group is which, but you can have a look now. 1, 2, 3, 4, 5, 6. The idea is that because after incubating and placing them into the microplate reader for recording absorbance values, you're going to actually see starting, or you may see differences at the baseline or initial few time points for across the groups, even with the control. So I just want you to bear in mind, there isn't an erasure. Remember, between the three, you guys are going vertically, but you're also following through the different components, which you see in the lab manual, but just make sure that you align them accordingly. I'll do the blank, hopefully, and you guys will just be able to take care of, or do as much as you can, I would say, within the next half hour, and we'll discuss a few other things when needed. I'll say another thing, as you can see on the slide, we don't have to incubate at 37 degrees, so please just pay attention to that when you're actually doing resuspensions or importantly, resuspensions and centrifugation. There should be spinners or centrifuges here in the middle bench and one on either side, so you guys will share accordingly. As well for methods updates, they're based at now 5,000 RPM, not 2,000 for 5 minutes, so bear that in mind. You'll just update it if in your methods if you don't have that included. Let's see. As well, let's see. Yes, so you can see some absorbance readings. I'm going to be doing, I'll tell you some other details when needed, but I just want you to highlight that when you are at the moment of spinning down um, your, um, your cells, make sure that you have your pellet and that you don't obscurely take it out when it's not supposed to be, because that can actually, deter the quality of your readings and when you place them into the microplate for further analysis. Any questions? So, and as well for the OD readings, I'll let you know. We likely will have to essentially use a smaller amount for Staphylococcus epidermidis, or actually, I'll let you know how they begin. In the meantime, you guys can get started. Exercise 1. You guys should be done in about half an hour, and thereafter be able to place it into the microplate reader. If your group is not done, exercise 1, I highly recommend getting ready at 10 a.m. Exercise 2, so that way you guys can flow readily and complete both exercises by 10:30. Any questions, please? Yeah. Remember with your Bunsen burners, make sure that you keep them on when you're touching any bacteria. Make sure you need it. Yeah. This is another point, folks. You guys have saline too? Okay. Just before you guys continue, just label your plates for NH. You guys have plates. Yeah. You guys can label your plates, and I'll keep coming back to it. You can also grab it like that. Here, like that. Here. Yes, sir. Girl. But I'm gonna, okay, I'll just clarify a few things. These are the tubes, the saline tubes that you'll be using for your cultures. So if you have a look on the red trays of your tubes with Staphylococcus epidermidis and E. coli, what you need to do is the following. And bear in mind that you are using the amount that you need. Like for example, following a septic you're going to be adding in, folks, only half of this to each of your solid bacterial cultures in these tubes. Because essentially, when you're swabbing, you want to have a thick, concentrated serum or amount of the colonies or bacteria. So just bear in mind that you're pouring in for the saline tube. You're only pouring in half for each tube. So that way it should suffice. And hopefully you won't need extra. Just follow a septic technique, but make sure you don't add it too much, otherwise it will dilute and essentially underestimate or limit your reading. Any questions? Feel free to ask. But you have the tubes that you need here. Your solid, liquid agar tubes are here. Bear in mind, whoever's taking H2O2 now, this is for the catalase test. Do not use this if this is for your actually exercise 1 because you don't know the concentration. So let me just go over something.
No.
So to clarify, folks, you have the following with you. You have Staphylococcus epidermidis and E. coli, and you have 3 different types of germicides, disinfectants versus antiseptics. Antiseptics are essentially germicides that are used in living tissue, and disinfectants are on surfaces like 70% ethanol on your bench tops, right? And antiseptics exist when people are getting medical treatment, let's say from different bacterial infections. So that's something that you want to consider. Now, that being said, groups 1 and 2 will be focusing on bleach. So you have the same 50 ml conical blue tube. It's a larger one that you guys will be sharing for across the 3 different concentrations. So bear that in mind. But group 1 will be doing E. coli, group 2, Staphylococcus. Now for groups 3 and 4, you guys are doing hydrogen peroxide. Please bear in mind you're using the right one, the larger thick tubes for the 3 different concentrations. And groups 5 and 6, you'll be dealing with ethanol. Now, before I end off, remember, odd groups, i.e. 1, 3, and 5 are dealing with E. coli, and then even groups are dealing 2, 4, 6 with Staphylococcus epidermidis. I want you to think why we're even doing this. Think about your unknown assignment, or think about lab 1. What's the difference between Staphylococcus epidermidis and E. coli? Which one, Ashwin, which one's gram-positive? Is it E. coli? Staphylococcus epidermidis. So just bear that in mind, folks. E. coli is gram-negative. We know that. Differences of peptidoglycan layers and thickness is very important because you guys are going to see today, and hopefully in due time from the absorbance readings, how they impact essentially your results. So that's something that you want to distinguish for the upcoming exercises as well for today. For the catalase test, for essentially the antiseptic, antimicrobial susceptibility test, if possible, but as well as the cephinase test, which we'll talk about, and you'll see the other materials in that PL bin besides you. So just bear that in mind, folks. You guys can continue. Keep prepping, and if at 10:10, if your group is not done or not complete exercise 1, have members of your group focus on exercise 2, and I'll be the antimicrobial susceptibility test, which I will talk about now. ODs for spec 20s, you don't do. I'll be doing that. You guys don't have to worry too much. I'll be taking those recorded records. Yeah. Any other questions? Okay. So you see that green print? Yeah. In this case, we can take 1 mil, means it doesn't hurt, but it has to take time overall. I would say though,
it's like
so I would keep because I'm going to be saying, I think positive, negative, and 25%. Folks, so your exercise, your mission for exercise 1 is on the microplate reader. Spec 20 results, like, don't worry about that. I'm going to be presenting them to you in due time. I just want you to understand, though, for exercise 1, briefly, once again, you have the tubes that you need. They're in solid agars for E. coli and Staphylococcus epidermidis. For your group, because you're either doing one or the other, bear in mind that you're adding in half of that saline tube in the green tray. Please, folks, add in this saline tube. You should keep it in your bench because you'll use it also afterwards. Just make sure that you use the right tubes because you're going to add in half of it into your solid bacterial culture tube. Meaning you're adding in half this amount, pour it in, mix it, and that's your way of resuspension. You can, there's other, you guys would have other apparatus, but it's okay to just use the column. Another thing, don't pierce through the agar. Just essentially use the saline as a way of essentially concentrating the bacterium present in your tube. Does that clarify those questions from the measure? Yes.
okay.
I'll be. So essentially, these two last two, but you still should be able to use another concept. I'll be doing the OB reading soon. So just keep going with the material, with the method, or with your flowchart.
Yeah. This is each of the files. What are we putting into the microplate reader?
Oh, so you are using this to cut up for the upcoming tubes. So you have other tubes that you're going to be. Yes. Yes. But there are additional instructions from the manual. Once you're officially done adding in the disinfectants for 15 minutes at room temperature, once you've done that with disinfectant and putting down your pellet, then with the liquid broth, you'll be adding them into here. So like the microphone. So technically it's yes, ignorant. Yes. How do you want to? If we're looking at 430, that's 200. So this would be that's 1,000. Or leave it 1. Okay, so this is 1 million. Yeah. Somebody uses this as oh, you're back here. I just want to make sure. No problem. Yeah. That's correct. You can use the pipette tips for determining volumes. You don't have to use these glass pipette tips. It may make it easier for you to essentially transfer.
Yes, yes.
100 means 1,000, right?
You
want that? It is. That's for
Yeah, the pellets are supposed to form in the centrifuge.
Yeah. So remember, you still have your centrifuges set for 5,000 at 5 minutes, 5,000 RPM. So just bear that in mind. It's an update from the message in your manual.
It's not effective.
Have you already taken out a few or?
What?
Okay, that's the one thing about the microplate.
That's cool. If you need to, you can.
I'll just say these. Okay.
The thing is, I've only been given a reserved amount. So what I would recommend is try to use be mindful. Thank you so much. Keep it up, you guys. Keep on.
Yeah, that's.
One moment.
Fine, your bubbles will.
Give me a moment, coach. Okay, Zara, what is the question? This should have containers at the end like this. Can you actually distribute those text like these papers? Oh, yeah, this goes.
So we're blotting out the our culture and dipping it in the saline. Okay, so we're blotting out our culture and dipping it in the saline. And then we'll take
Or you could essentially,
It wasn't.
you don't
Right, yeah. I changed my class.
and then Did you find these ones? Okay. Give me a few moments, folks. This should be done clear. Okay,
I know. We're just centrifuging for it. We put one going each.
if you guys find a bin, they don't have to. You guys do. Or ask if you want. You can also do else. It's probably need extra weight. Thank you so much. Yeah. Yes. I recommend. Yeah. I recommend also confirming any other groups that need it. Folks, when you're spinning, just bear in mind a few things. Have all of you begun spinning? Yes or no? Okay. Okay. Perfect. So let me just clarify that. Your next steps are going to be discussed shortly as I do the performance readings. make sure that you're doing them as sets of two, because of the fact that we have only 3 centrifuges. So you can make use of spending the time together, not wasting and waiting for an extra 5 minutes to do your first spin. That's just my tip for now. If your group is already midway into this, they could start beginning with the antimicrobial susceptibility test. I'll talk about a few things soon. Just give me a few moments.
On 5,000.
Yeah, so speed 5 is 5,000. Speed 5 is 5,000.
Is the laptop still there, John?
Should we call him?
It's okay. It's okay.
I don't think Russian is so excited, but guys, Canadian. I was like so familiar. The what? I was like, I'm very delusional. I'm not trying to do it.
Yeah, so I would just wait a second.
Okay, I was like the same as what's in their part. It's like I don't recognize the face.
So I'm gonna switch the yeah.
Balance.
Oh wait, we can put our sleep.
I would wish so.
Essentially, by the way, is there another balance? Like keto?
It's all about the learning problem.
You guys look great. So, okay.
Obviously.
Yeah, maybe don't burn your face.
Physics one. Wasn't this question? Physics and companies.
I didn't think of it.
I'll just remember. I didn't know what the guy short my GPA.
So we're going to be around 3 to 4. Then we're going to be
Should be good. In the meanwhile, could we do that?
in the meantime, I would say get ready for the melting another Hinton agar plate for essentially labeling them. getting prepped for exercise 2.
One's the bio.
Yes, yes.
Okay, so this will be the like 3%. Okay.
I was one to stop.
Okay, let me just answer some questions. Update from everyone. How's it going for exercise 1? Are we done the first spin? Yes or no? We are? We're into the first spin. Okay, okay. Are you also done? Okay, that's okay. For the incubation, yes. And by the way, that will be done at room temperature, not at 37 degrees. So please do not place them into the incubator. You don't have to. Now, that being said, I'm going to go quick to remind you guys that at around 10:30, all groups should have added in. Folks, please pay attention. All groups must have their tubes added in as shown here for each group. Blank, don't worry about, but for tubes B to F, they have to be added in into the correct wells. Please make sure you're following the order. Positive control first, negative control, and then your low, mid, and higher or highest concentration for your germicide. Please arrange your tubes in that manner as well, so that way you'll add in 1 ml each time readily here with a different tip each time. Except for, actually no, each time would be best. Now I'm gonna go straight to the antimicrobial susceptibility test for those that are on it. Yes. Negative control should be saline. Wait. Sorry. Positive, negative. Let me just confirm. Your negative is the one that essentially would not have your germicide, i.e., it would have 0%. So like have essentially, I am assuming everyone did make 5 tubes, right? Yeah, so your tube C essentially, the one that is your negative control, doesn't have the germicide, including bacterium. I'm just thinking about anything else. You should show it to me. Yes. Show it to me for the first time. Yes, that should be okay. As a negative control, I'll be brought. You have your pellet resuspended, yeah? Okay. What is that?
This one is 4.3. Those are other ones. I love how it gets less and less out.
You have another one. Oh, your negative control. Yeah. Okay. Perfect. So if any of you are having difficulty, you can pipette up and down. Let me see if I can get you guys. Sorry, I'm gonna grab these two.
These are not there.
These are for waste. So after you use them, come with a pipette tip top. You guys would have a pipette tip top.
We use those.
Okay.
I thought they were new.
This is I would recommend though, like from the tips that you guys did select though, because you know we have the question I have is that any of you use
No, they also.
Even some of the ones that are don't.
Yeah.
because I saw one that had when you hit these close, always make sure that one minute. Sorry folks, because I don't want to go behind schedule. Let me briefly go through exercise 2. So pretty much, you're gonna make plates that are labeled. Folks, by the way, is your question exercise 2 related or? Okay. You want to show me your pellets or?
Before we added the
Okay. You can still do it. Yes, that's okay. Do you guys still have enough ethanol?
Yeah.
It's okay. No problem. So, for exercise 2, actually, any other questions regarding exercise 1? In about a few minutes, I'm going to give you your, I'll talk more about exercise 2, but just make sure you have your plates labeled. Make sure, just like what you see in the lab manual or on the slide, that you have 5 different quadrants, 5 slices of a pie on your agar plate for E. coli and Staphylococcus epidermidis. Yeah. You would have labeled them? You guys just want to make sure. Hopefully, you guys have. But make sure you put the tips to other places.
Very small.
Give me about 2 minutes, folks, before answering questions. I want to give you guys. Okay. You're gonna get the screen. 1 minute. I just gonna share some details. So folks, I'm going to show you now the results to describe a bit of what you need to do for calibration purposes, or just to be aware for your whole lab for this week. These are the initial absorbances for your bacterium of interest. Now, generally speaking, as I talked about earlier, you have different starting absorbances or turbidities at baseline for both cultures, which is why we want to be able to distinguish them and the trends that they would grow towards or be impacted by. Folks, let's pay attention with the different concentrations of your germicides. Because the absorbances are relatively similar, what I recommend is just going through today's practical in the same fashion. Don't worry about additional steps. But I'm just letting you know, record the absorbances I have here, 0.73 and 0.85.
That's for experiment 2.
Exercise 1.
Exercise 1.
Remember about spec 20's OD? Some of you were asking about that. So I did that. I'm going to be taking care of the lab. We're completing a few other things. They're fine. No, they're already blank. That's okay. No, like essentially for spec 20, you have to blank it in advance. The blank absorbance readings you're gonna see with the microplate reader. You don't have a separate, because when you're blanking, you're trying to reach 0 for absorbance. So yeah. Yeah. Spec 20. Give me just 1 more minute, folks. I just want to complete something. But keep it up. I'll just be helping folks in the next 2 minutes because I need to clean up the space as well. Cleaning the line to you guys. And if it feels intense now, the good news is that it's going to be much more calmer soon. So don't worry, you guys. I don't want you guys obviously to be stressed at any moment in the lab at any time. But I just want to let you know that it'll be hopefully easier as long as you make the right. As long as you do things correctly. Very intense.
1,000 is 30 equals 300 foot.
Could you repeat your question?
Oh, this 30 on the 1,000 hit 300 well.
Which sector are you?
1,000.
You looked out.
In there, we want the 300 well, so.
We're treating this cotton spots or clean off the lady grocery. With exercise 2, treat the entire plate.
The entire plate before we added this. Yeah. So just let me just answer any further questions in a bit. But remember, your plates should be labeled. Label them with your group, label them with the bacterium, label them with the exercise, label them as we've discussed in the past, and make sure that you have, folks, just a tip that I would highly recommend for your plates. Make sure that you have 5 different sections, like a pie. It can even be easier done.
Treating the plate like we streaked the plate last time, or are we doing each section?
No, so the advice that's given is that you do it 3 times, 1/3. Think of it like spread plating. But the thing is, so like let's say you've already labeled your plates, both of them, with the E. coli, for example, the exercise, your practical, for let's say P8, group B, group 2, and labeling, of course, all the letters A, P, T, etc. Now, to answer your question, use if you still have from the saline tube and remaining residual bacterium, you could pour it back inside that tube, or you could pour it into your other tubes, the other set of 2 tubes, which would have E. coli and Staphylococcus epidermidis. With that in mind, what you're going to do is essentially use that cotton swab, get it like concentrate your bacteria, or get a good amount of it. Do like what we've done in the past, but all around. And then turn it around 1/3. Repeat the same process.
The second time.
Where you're dipping. You can dip it.
Just to use nobody what's already on the plate.
Yeah, let me think. Can you do the L rod? And actually, to clarify, great point. You use the same swab because technically you would actually have to use a Bunsen burner to flame it. The point is that you do the same, repeat the process. Once you've essentially added in your bacterium, we'll discuss further what next steps are to be done. But I would recommend having other members of your group start getting ready to prep for the microplate reader in the next 10 minutes.
What's the negative control?
It's going to be, I believe, the LB block.
It's the LB block. Yep.
It is specified in the manual.
Probably.
So spin that incubator.
I use the one that is for your Actually, yeah, start off with one. I'm gonna just finish this and I'm gonna see what I can get for you guys as extra.
As you mentioned, the residuals.
That's correct. The issue is I'm just wondering if you guys have enough. Give me a few minutes. Okay?
We're finding the discs for the antibiotics. Okay, is it okay if we wait between street plating and putting the discs?
I need to ask, how many did you get, buddy? One? I want to know if you specified about, because technically you guys need more. Each of you. I just want to give some context, folks. I want you to have, you guys should have the materials that you need, and I'll do my best to give you if needed more, but you may have limited. So just bear that in mind that you're controlling with how much you really need.
Yeah.
So like what I've done actually, they're in the entire surface Okay. Can I just ask for an update how everyone's doing? Exercise 1, are we in the incubation phase? Second, so you're done the incubation. At room temperature. Like you're at the second step. Continue. Perfect. That's okay. That's okay. I'm gonna talk about that in like now, but I just want to confirm everyone else is doing good. Groups 4 to 6, are we done the incubation? Are we in the incubation phase? Perfect. Likewise? Okay. Group 4, are you guys incubating your germicides? Yeah. This is hydrogen peroxide, right? Yes. Okay, so now, folks, as you're waiting, remember 15 minutes, have a timer at room temp. I'm hoping by at max 11, and I'll say right now to make it easier, 10:45. You guys should be prepped to begin adding in your bacterium or the wells the tubes into your wells respectively. When you're there, just let me know, or I'll give descriptions here as a reminder. Follow with the group orientation as you saw earlier. Yes, so exercise 2, 3, and 4. 5 is just on slide, so I would just say once you're there, you can begin prepping for your post lab, write down your results, observations, and project exercise today. This is sterile water, by the way, if you need it. I'll talk about it. So, very important. Please return back all tubes, all discs and vials back to their locations because they have to be used by other people accordingly. And obviously, we don't want to throw accidentally anything into their basket. This for the second test. This is exercise 3. You'll find here. Before I go to there, exercise 2, as many of you are wondering, we have a failed tray with all 5 antibiotics. Each of them should have 5 vials or small tubelets, and the idea is, I think some materials have been misplaced. Anyone finds the tweezers? There should be tweezers. Forceps. Yes. Okay, so I just wanted to let you guys know, you guys are doing great. The thing is, there's some materials that are missing here. And I apologize. Please understand that I am not in full control of the materials that are here, many of which is available from the last time. So if there are times where things are at pause or wages, please understand. I actually have to get you guys new forceps because these are supposed to be sterile for this assay. But the idea is very similar. You'll be able to take it out from one edge, just push, as it said in the manual, push from one edge the disc that is on the top side, push it, and then take it out from the other end. And essentially, I'll show it to you guys. It's difficult now, but when you're at the plate, we'll be adding them into each plate, each section per plate. When you're done, when you're done all antibiotics, please return them back for exercise 2. Exercise 3 is 7 test. You're doing them on the glass Petri dishes, which are down there. And an example is that you are essentially adding in only, and then 2 of these, by the way. Marvin, and I believe this group has one as well. So this bench could share accordingly. This bench could share this one. It's very similar to what I said earlier. You're pushing it from one edge and you're taking out on the other side. So you'll see, like essentially, because it's straight in the middle, you want to be able to take it out from one side. This, you just push it on the other, push it out the other side, and you pull out accordingly. Push and then pull. And then you place it for the 7 test 3 in a glass Petri dish. And then you'll have your spot drop of your bacterial cultures with water under control. Water's here. For exercise 2, I just mentioned it again, but just to repeat, you're pulling out just like these vials. You push out from one edge, you pull out on the other, and you'll separately with sterile freezers, which we'll hopefully get to, essentially press gently onto the different sections. And then try to get them in the middle of each section of the 5 that you have, so that way each plate will be able to represent the different zones of inhibition, which is good for next week. Technically, these are not like they are not so they're not bacteria essentially, which is why you don't have to clearly, but I would recommend. Exercise catalase test. If you guys are in the midst of working ahead, you have hydrogen peroxide here. You have glass slides well available, and simply put, you're doing what you've done in the past of bacterial smear. But really, it's very simple. Adding in a swab of each bacterium, Staphylococcus epidermidis, and of E. coli, and then you're going to add a small amount of hydrogen peroxide on each one with the plaster pipette. You can also use a very small amount, and hopefully you'll see results in real time. Just remember to record results as you do for the figures that you have for your Bible counts and for any other results for the translation Japanese test. Thank you so much. If there's any questions, feel free to raise your hand. It is possible. Wait, no. Then they will come for thinking many of you have them already on your benches. So let me just show you guys what you need to do. So, yeah, that's a great point, which I'll have now, folks. Let's just point out a few things. Earlier this week, you guys would get sterile tweezers. However, what you really need to do now is just like what you've done for the L rods. Added 90% ethanol in a glass petri dish down there, and then essentially flame. Let it cool down, and then take out one disc at a time into the appropriate locations. You could be gentle in doing this and just pressing on the top, but the idea is, I'm taking it on one edge. Remember, I said push, so sort of push, and it's not in the middle any longer, and then pull. So it's gonna. I don't wanna chip it out for you right now, and obviously I'm not at your plate, your bench, right? But the point is, you want to just push it. You have different ways of doing this, like how I just did now, and then you should be able to take it out accordingly, and then pushing it down. I'll just take this one out as I test it, because it's technically actually, if you want to sterilize this, if you find it difficult, then just take it out, take up the next one. Yeah.
Okay.
As long as you're using it on the same culture.
Okay.
At the very end though, do sterilize your tweezer with 90% ethanol. So that way you can reduce contamination with other species and also for other practicals as well. The tweezer? Each one should have one.
Should center.
Okay.
If you doesn't have a tweezer, just go straight ahead to the catalase test or the the catalase test. It's more simpler. You have slides available as well. Let me just read it here.
And all the way?
No, it's not good.
But this
That's not it.
Is this side one?
Yeah, that's true, but I would recommend, yeah, okay. Technically, those are supposed to be autoclave. I don't know if they autoclave those. Let's see, I'll ask if there's any. Well, if you need to laugh, please let me know, because I need to make sure that you guys are all safe, of course. Now, college-wide, if you don't have a college, that's okay. Just take out at least 200 microliters, or actually 20% of your specimen, of your volumes, and you can be suspended accordingly. Any groups waiting for their last spin? That's okay.
That's smart.
Yeah, okay, good. How many groups need tweezers?
Yeah.
Here. Sorry for the slide.
Ask questions in the meantime, as well as, so once again, slides are here. The materials that you need for exercises 3 to 5 are here. Let me just make sure. The last test isn't here yet, so I'm just hoping that you get don't worry if you're waiting in line, try to continue with the catalyst test, and you can also thereafter get ready for the other tests. Let me go straight to your generation time assignment. Well, let's see. Okay, actually, are there questions for the FLR, formal lab report? Plaque assay due March 8th for many, like on 10 p.m. Is everyone okay with the plaque assay, page assay, formal lab report? Both results and in your, yeah, so bear in mind that you're being as descriptive as possible for your FLR. Yeah. But, have you guys ever used tweezers yet? Not yet? Garvin, can you give it to them because they haven't done it? I know you guys have done it, right? Oh, you haven't? Oh, okay. Okay. If groups are done with the tweezers, please let others know so that has done it before. I'm going to see if I can find them for you now. So how long until you guys should be done for exercise 1? Can I get an estimate? seconds.
I would take
About
10
10
minutes.
minutes?
For, yeah, like,
I thought you have one more.
one more.
So about 5-ish
I would
minutes?
say 5.
5 to 7. Okay. Groups 4 to 6, are you guys good for timing for exercise 1? Do you think you'll be done in the next few minutes? I'll give you guys 10 minutes. Or to be more precise, 9 minutes from now to be done exercise 1. Or, yes. Are you done? Yes? For your plates, 5 different antibiotics, you've added them. Yes, I would recommend giving them to others. If you, yeah, I would recommend giving them. You will need it afterwards, but yeah, it's better for others to use. Okay. Stephanie's test. Anyone else having need of a tweezer? I'll tell you, you needed a tweezer, right? Perfect. Great job, you guys. Great job. Okay. 2. You're done? Okay. Remember, folks, please place back the antibiotics into the appropriate places. The discs need to be in the right spots. And for a bit of prep for your post-lab, start to look into what you predict would occur for these different results.
When you do the 7 days, you do them heat
I would say count this one and count this one.
Because it's to use only one type of table.
Yeah, that's okay. But if you could, yeah, that's okay. Provide just the details, all the details that you need. But actually, yes, the table works perfect. No problem. No problem.
I'm doing exercise
The exercise.
with the
Yes.
exercise 4. Sorry, my brain's not
It's
working.
okay.
Do I just
Okay. So hopefully, I'm just going to walk through a bit. We've talked about the susceptibility test. In a week's time, hopefully you'll see the results of how they differ for the different antibiotics treated for your both of your cultures, Staphylococcus epidermidis and E. coli. Now, exercise 3 is your beta-lactamase test, also known as your cephinase test. It is in those longer rectangular white tubes. Please bear in mind we only have 2 of them. You have to return them back to the right location, back into the BD boxes. Now, for those that are wondering, these next 2 tests are important for not only categorizing the different bacterium, but also being able to apply and understand molecular biology and how essentially these bacterium can be impacted through medical treatments. So for example, we know that beta-lactamase is the enzyme that breaks down the beta-lactam ring in different bacteria. Those that exhibit resistance to the penicillin family, i.e. antibiotics that are able to cleave the beta-lactam ring, these are essentially ones that will identify of not having a rapid change. Having a rapid change in color with the discs that you have in that in those vials. Nitrocephin is going to react to essentially be similar to the beta-lactam ring, and under oxidation change from yellow to red. And so that will be an indication for you guys for your post-lab quite readily to record the results and to determine if, for example, Staphylococcus epidermidis has a beta-lactam ring or not. Sorry, if it breaks down beta-lactam, wait a second, no. If it essentially has a beta-lactamase enzyme to break down the beta-lactam ring, and or if it's negative. So just bear in mind that you understand if the test has shown positive results or negative results and what that would be inferred for. Either it's beta-lactamase positive or negative. Give me just one moment before you guys go forward. The catalase test is similar in giving you a positive or negative result with the same bacterium that you're using from before. You use the cultures that you have available, but you're using the hydrogen peroxide in the thinner blue tubes. And so you'll eventually see rapid bubbling or slow bubbling and thereby be able to depict if it is abundant in catalase or not. So these two exercises reflect the propensity or how much of an enzyme is exhibited in the bacterium. Either they have a lot or little of beta-lactamase, or they have a lot or little of catalase, and or are positive and negative for the beta-lactam ring. So just bear that in mind, folks, for being able to rather cleave or react accordingly with certain products in the environment or within other cellular systems. Yes. Yes. That's it. That's it. That's okay. Does anyone need tweezers, by the way? Now, can you just about 30 seconds? Back up on the line. I will show you guys in a moment. Folks, I just want to make sure, is anyone else almost done exercise 1?
Almost, yes.
How many minutes?
5 minutes.
If anyone's done earlier, please come closer so that way I can show you what you need to do, especially those that are going to be pipetting in, because you guys will be needing to be under supervision for placing it into the microplate reader, just to give you a walkthrough and make sure that you put in the right volumes and tubes into the correct wells for your row. You guys are which okay, so you guys will be this positive, negative. Perfect. So you guys are ready. Guys can begin with. I'll do one just to make it easier for you guys. To start. Because believe it or not, you guys are primarily in the lab, but sometimes I come in here. I would say, I would just say, make sure If you treat the immune system, treat that well, and then when you press into the wall, make sure you have the wall. Because you don't want to, sometimes if it's sensitive, you don't want the drop to go into another wall. And that'll be killed by the
No worries.
Yes.
I think I already. No, I think I already.
One missing the cube that they need. The last time that they need to come back, so let me know that. You guys want to.
Oh. You're gonna just turn the timer. We're gonna push it. Should be good though. 5 minutes. Okay.
Having classes.
Yes.
No, 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 I'm.
So, on this one,
If you guys need extra tweezers, we have them here. A bench more for exercise 3 as well.
Yes.
Remember to resuspend your tubes when you bring them up here. I do when you guys are done this, I'll try to go through other points, including, let's see.
We should be done very shortly, actually, in the next 5 minutes. We should have that.
Who has a Stephanie's test tube? Who has Stephanie's? Who has Stephanie's? Anyone have Stephanie's? Oh, it's okay. It's likely, it makes sense that it would have run out. In Parker, everything okay for your group?
Honestly, I'm a bit behind for number one. Also, I'm not sure if I have time for the hand washing thing, but aside from that.
Don't worry too much about that. Sorry. Calling swap? Calling swap? But place it on the slide first, for both of them, separate and separate. And then add in the hydrogen peroxide with a factor of like a small amount. Just use one because it's easier. You only need two bottles. Like you just like how we did for like essentially you should have enough space, I would say.
Plates for exercise.
That's on slides, so don't worry. That's I'll talk about. So if your group is done, your group you were the first group. Okay, so record your observations for exercise 3 and 4, because that way you can help prep for the post lab. If there's any extra things that you're missing, let's say from what I've talked about for previous options for post lab or free lab, like basic exercises and then you can include that as well. This can make use of the plan that you have. Any final questions? Keep it in.
This is, sure, it's just nutrient, correct? But it is some of my yeah, we have to put into the next one. Okay.
It is actually. It is.
We should have started with supernatant.
LB broth, no. For the negative control, like it's so for this I could talk about that in about 2-3 minutes. I want to make sure everyone's on the same page. One moment, if you don't mind. So there was a group that asked for Stephanies. Can that group raise your hand? Or the group that has Stephanies with them? Yeah, who? There was a group 1. Ask group 1. They should have it. Sorry, group 3. Group 3 has. Yes. The catalase. One moment. Okay. Carlos, just to answer your question, go with LB broth, resuspend your pellet. Resuspend, so you remove the supernatant. Yeah, so, no. What I mean is, sorry.
In this one? Oh, okay. Not doing that.
I think, was it you go something? There was another group. Five is actually as well, that's fine. Okay, perfect. So, yes, I would say give it to the other. Okay. Did you get it, Zara?
Yeah.
Perfect. How's everyone? Okay, so let me talk about. That's okay. Was one doing okay? Comparing the two, was one different? Okay, that's okay. That's good. So think about then, because catalase is an enzyme. Think about then comparatively, how much one has more than the other. Like which one would have more? No. Essentially, one is higher and one is lower. Yes. Well, did you see that from the seven? Yeah, from the 70s test. Okay, that's a good sign. Take photos if you wish. You can draw them out for your observations. Remember, folks, into the Wascadine, yes. Actually, it's better in these tips. You see you see that bin? Yeah, that's the one. Are you done? You're done, right? Yeah, perfect. Then it should be okay. Perfect. So not yet. There's a few things to touch upon. 2 to 3 more things. I just want to make sure everyone is let me pause right there. Sorry. Another thing that which people did you start already? No. So I wanted to should I put more because I just lost a little bit of this thing. You can Yes. Perfect. Okay. So if you can
Add more albumin because I lost some of mine.
well,
That's fine. Like this.
it's fine as
Because it's positive.
and I just don't want to get your diabetes.
Yeah.
Let me just show you. And then-
Yeah.
Reason why is because you may get it underestimated. So let's say it's your group. I'll let you. I just want to give you some context. Go inside. Then and do the exact same thing each time, and then you just dispose of both of them. You tip with the tip inside.
I think so. You need help? Don't forget me. Oh, yeah.
So, so just read this thing, all of them, I would recommend. I think the key is just in case, read the sentence. I'm gonna go over a few other things. Keep it up. Inshallah. Okay. Groups 3 to 4. How long do you need for exercise 1 to be done? Okay. Carlos? How long? But you already on. I thought you did your third speed. Okay. You should. Well, let's see.
We did this was fun and you got rid of the. So we had this.
And I just like.
It could be a small speed. And then you should be able to contribute peroxide, classify tests or the small.
Pasture is the one that was like.
Yeah.
Okay.
Or banana. Keep it up to that. Now I'm gonna go through the hand washing exercise in a bit, like in a minute or so. Okay, some of you are asking as well. And please, folks, I don't want you to be distracted. Please do not use your phones or leave unattended without letting me know. It is actually, important that you inform me as a TA that, you're going out to use the washroom, for example, or you want to take a photo, etc. But you can record your results as we've discussed thus far, right? For the Cephalase test, exercise 3, to the catalase test, exercise 4. Any even limitations and mistakes you may have done for the first 2 exercises, something you want to keep in mind as well. Now for exercise 5, before I move there, any questions for the previous 4 exercises? No questions? So exercise 5 is a hand washing one. Back in the day, I think this would have been done. Essentially, you see the sink as you come in, right? You have hot water, cold water, an intermediate warm, a mix of water. You have soap available at times and sometimes sanitizer outside. The idea is that some of you may be wondering, what's the best way to essentially, the best way for you guys to be able to clean your hands, right? Because obviously as you go in and outside of a lab, you want to be very careful, right? So what would you do? Believe it or not, one idea is, and often you would think, okay, use warm water, right? Because you're going to kill off your bacteria. Not necessarily, actually. And this is something we're going to talk about in week 7 or 8, about the different temperature ranges that bacterium exists in. But as you could see, there's still bacterium visible across these different conditions. Yes, in different amounts, but something to point out. And if you look here, with the questions that you can answer for your post lab, look at first the morphology of this slide with the different plates available. Also, group 3, when you're ready, please come in as soon as you can. I'm just looking at the rows left for the microplate reader. But so you see different colonies, different shapes, different smears, right? You could see it on the slide. Hopefully those on the left side of the class can see as well. But you see different morphologies of them, likely different cultures. So cross-contamination, if you're not too careful, can occur. Which is why even you may want to consider between major stations or major steps of exercises, especially when you're going to a microplate reader or to your plates, to rewash your hands accordingly, because you may have touched your bench top, you may have touched other things, your pencils, etc. So, or your phones. So just keep a little bit of that in mind. But you have a control, right? As we've done for the plaque assay, as we've done here even for today, and the blanks as I've done, alhamdulillah. So you'll see the results accordingly in due time on Quercus. But now if you look at this slide, I know. And so the alternative is I was going to talk about plant biology. So essentially, you have what's called fat, right? So saponification for those that have taken orgo. You actually can theoretically use oil and make fat. So that's a separate point, but make soap. But that being said, we'll go to the soap soon. Before you guys leave, but I just wanted to highlight, based on the different treatments or conditions that you see here, hot water still appears to have a lot of bacterium present. Cold water seems to have even less so. And then the addition of soap helps to control think of it like this, bacterium that have capsules, right? Because they have a waxy surface layer, right? Like as we've talked about a lactis, if you recall. Parvin, if you want, you can even use those pipette tips. Might make it easier. But essentially, have the different conditions as you see. Sanitizer also helps well. As we talked about, ethanol is a type of germicide that you guys are looking at today. And you're putting it on the surface of your body sometimes, right, with sanitizers, sometimes on your bench tops. It serves as a disinfectant for you guys. So that's something you can think about to apply the different terms into real life. And the video shown here also provides greater detail and actually reflects what we are about to talk about here. So before hand washing, let's say you put your finger or your whole hand into a plate, or if you were just to, swab different parts of your body or smearing from your mouth, you'd be able to get an idea as to how much or how many bacterium are visible and actually could grow after time, over time, essentially. So before hand washing, you see 4 different clusters of colonies. Here, after pore washing, you see dispersed, the similar amount of bacteria. Here, the quantity of clusters. After proper hand washing, you still see some colonies, which folks, you should actually come to appreciate not all bacteria are harmful or deleterious. Many of them exist in our gut flora, right, and are beneficial for our bodies to survive. And sometimes you want to supplement them with prebiotics, like with yogurt, or probiotics as well, with like supplements as well. So those are things that you can consider and discuss if needed in your discussion. Because often you may get confused. Not all bacteria are harmful. Many of them that we've grown thus far are beneficial for different treatments. If you're into like medicine or pharmacology, then you would actually want to develop, let's say, E. coli to have a specific plasmid of interest. Let's say insulin, right? Or it could be ampicillin resistance for other selectable markers. Or like what we've done thus far today, being able to analyze, and hopefully what you'll do with the Muller and agar plates, exercise 2, be able to explore the zones of inhibition of different bacteria and explore how that could affect different treatments that you would give for particular individuals suffering different maladies or different concerns. So after hand washing, no drying, so not going under the hand dryer. This is what you also see. So think about the different sensitivities and growth factors or patterns that different bacteria have. And if you were to, let's say, isolate them to have a pure culture, as we talked about in that 3, and hopefully, or not 4, hopefully I appreciate that you will be able to isolate and derive certain characteristics of the ethanol. Usually they do not, depending on what they are, because the idea is high pressure, hot, vaporized air coming through. So you're going to have essentially that stream of air to reduce the presence of any bacteria. There is that potential that if you go up too close, you actually touch the surface of the air dryers and you get bacteria. But that being said, generally speaking, you're reducing the amount of bacteria that you're going to already started off with. Quick question. And then after you cough, right? So it's important to be mindful, right? If you often will cover your mouth when you cough, or you'll blow into a tissue when you sneeze, and different things to bear in mind, right? So you can see milieu, that's like many essentially bacterium that exists across colonies, shapes, and arrangements. Let me go straight ahead to the generation time assignment. Any questions at all? Yes. Both are here. E. coli and Staphylococcus epidermidis, the ODs for the baseline at spec 20 for the spec 20 is shown here, meaning that you don't necessarily have to equilibrate the absorbance readings. I do suggest down the road, if you like get into, let's say, med school, grad school, where you have to put specific details in and account for these controlled variables. Actually, oh, okay, sorry. I thought that was liquid. I was gonna tell you no muscular. No problem. That's okay. That's okay. Make sure you resuspend. Perfect. That's okay. That's okay. So, that being said, we're now into the generation time assignment. I posted the material online for you guys, so you should have everything that you need. If your group is also done, you can, as I talked, clean up your benches with 70% ethanol. If you're done with your viable counts and your plaque assays, with your data, you can share it with me over email. But I just want to highlight for the generation time assignment, in your notebook, you could take the notes of what I'm about to share in due time. You have the materials, right? You have the materials that you need. I posted them on in our module section on Curtis. You have semi-log paper. Yep. Okay. Try to go in straight and then aspirate out. Perfect. Keep it up. Now, with regards to generation time. Okay, so imagine this is your semi-log paper. Does anyone have it with them right now? I can show as a good example. So let's say this is your paper. What you're gonna need to print out, by the way. If you need help, let me know. But it's posted on Quirkus, and the idea is that you have the results that you need from the microplate reader on Quirkus for practical 8. You are going to look, and if you essentially look online with the guide that I provided to give you guys further instructions to succeed in the lab or the assignment, I want you to consider the following. You have your intro at the back, 3 to 4 sentences, 5 sentences max, or in the detail that you need to describe, as I talked about for the FLR, going from the general to the specific, i.e. talking about generation time, or the different phases that bacterium grow in, or go over throughout time. You can talk about the lag phase, the 4 phases that we know of. Log, stationary, stagnant, lag, and the death phase or morbid phase. Now, that being said, you talk about this, you talk about your purpose, the objective of the lab, your research question, and then you go straight to your result. Your results are going to be on both sides. So to highlight this, you have 4 different curves, and this is just to highlight it here, that we're using semi-log paper, meaning the increments that you see here, please pay attention, folks, because this is very important. The increments don't go up by like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. They go up by a multiple by 10, meaning that your first major increment, after going, let's say, I'm going to erase a few things, or just show it to you here. So, sorry, that was too loud. That's good for you guys. But let's say this is your starting of the graph. It shows 1 here. You can see. Yeah, I'll move accordingly. But I just wanted to show that it goes up like this, and it further goes down. These increments are like your exponents, meaning that, let's say, because it's going up by 10, right? So if let's say this is 0.01, 0.01 for your absorbance readings, within a range of this or around it, you'll go to from 0.1 to 0.1, then to 1. And so I want you to just keep that in mind for your readings. And essentially, that's what you're going to be looking at as you take detailed notes or essentially plot out your results. Let me see if I could say this in summary. Yes.
So do you place all the T0, T20, T60 into one paper or separate?
I'll talk about that. I just want to also see if the microplate reader is done. Are you guys done?
Is it okay if there's like not much stuff on the bottom? Trying to resuspend it.
Oh. If you have difficulty, folks, this is what you can do. You suspend it should be okay, you know? So really to highlight, actually, let's see. I just want to wait for this final reading and then I'll talk further because this is timed for our lab 2 weeks from now at the beginning of the lab. So I would say if you haven't done the plaque assay assignment, FLR, focus on that. But you have the results that I need as well from you guys for the plaque assay and for the viable counts that you've done thus far. You've done only 1 minute, right?
Most of the school.
Keep it up.
I appreciate your enthusiasm.
Your result. So what I mean is, great question again. Give me a moment, but to highlight this, folks, yes, we're gonna be putting the, okay, you had a question about data. Plaque assay results and viable counts, please send them to me via email. If there's time, I just want you guys to be able to go through everything with ease. I have to put this into the microplate reader.
Do you want the results? Just
your, so for example, like shown here, you show me, you can give me your colonies. I have a feeling someone's recording me. I don't mind. But essentially the colonies essentially are the following. You put your cell density. So let's say it's your 217 colonies at T30. You're going to be giving me essentially for your viable counts the total value. So it would have been like what I showed earlier, 2.42 times 10 to the power of 8. That was for PFUs for the plaque assay assignment, FLR. Generation time assignment, just saying that it's CFUs, right? Colonies forming units. So you guys could do that for all of the temperatures, all of the time points across the dilutions that you have. If they're outside of the range, for example, most of them appear to be, and let's say if this one is the only one that you're able to grab, then essentially still provide that information for me so I could essentially provide that all to you guys for the group so that way you can complete the generation time assignment efficiently. I'm going to go through the results. I just want to finish this. Give me a moment.
You said the viable counts and the plaque assay results for who's great.
So, I think there was one there and then side. Okay, what was the question from this side? Was there a question from you guys or no? Perfect. Okay, sorry, Saad. Trust me, I'm not trying to. I don't want to like. I worry that I may be unjust at times, so I don't want to like unaccount for anyone. Now that being said, let's say you're looking at your microplate reader. This is an example. So for context, the germicide microplate reader is now on, so it's going to be taking absorbances every 15 minutes. So now you guys don't really have to worry too much about that. We're ahead of the game, alhamdulillah, Alameen. But I want us to make sure that you guys are done the generation time assignment as soon as you can. I've given these results or these pointers for you guys to help you all succeed. I just really want to make sure that you guys are able to complete this easily. Have a look on Quercus. You'll see the results from the microplate absorbances for the generation time assignment. All of them are for OD 550, so make sure you include that as your units. But the idea is, have a look through the instructions I have here. You have essentially, our lab is practical 8, practical 7 is doing 30 degrees, and we did at 37 degrees for testing the array of generation time for E. coli. Now, I want you to pay attention in that you have the results here. I'm going to actually show it to you in case those that can't see from this side. The following. You have on Excel all of this provided in bold. I just showed it as one row for a sample, but you're going to redo this, folks. On Quercus from the results, you copy them accordingly, and this is what you're doing. You're getting your mean absorbances. You're gonna from essentially the first 3 reps, your experimental reps, i.e. the second to fourth well that you've done for each group. And that would essentially give you your mean absorbance value, and then you correct it. So it's like, as you can see, the mean absorbance is essentially the average of the absorbances of your first 3 reps, subtracted by the corrected, by the blank to give you this corrected absorbance reading. You do this for all groups, everyone, and then you should be able to get your lab average as the final value. You're going to do this for all time points, so from 0 all the way to 120, and you're going to do this at 37 degrees and at 30 degrees. So just bear that in mind, folks. Technically, for the microplate reader, you're going to be doing 2 different curves. For 37 degrees, plotting out the lab average absorbance values with OD 550 as your units, or 550 nanometers. And essentially, if I go back here, you would plot them out on the semi-log paper, following through from what I've said thus far. So let's say this is your microplate. You're going to likely see values that are similar to the following. I hope you guys can see. Please let me know. So the idea is that you have dots, you have your units, time, minutes, increments are even, i.e. 0, 15, 30, all the way to 120. And you just want to bear in mind that you're plotting them correctly, essentially, right? Because the increments are going by log, which is very important. So you just want to make sure that even the smaller increments, they're going to be going like from 0.02, 0.03, all the way up to 0.1. Just remember they're going up by 10. So the increments essentially, smaller ones go up evenly, but the larger increments are multiplied accordingly. So you're going to go up all the way, depending on what your data shows for the lab absorbance readings from the microplate reader. And essentially, you're going to plot those values as a curve. I'm going to repeat it there in case you guys can't see on either side. But the idea is that following your plotting of and using detailed figure captions or a detailed figure title with a descriptive reference, you're going to be able to make those 2 curves for 30 degrees and 37 degrees, plotting out the lab averages. When you're done that, and I'm just going to go to the PowerPoint because no, the time is all in one curve. So each curve is going to be based on time over either absorbance for the microplate reader or viable counts for cell density. That's a great question. So pretty much it's, as I mentioned, microplate reader, and viable counts. Yeah, I didn't talk about viable counts yet because you guys have the results and you're gonna give them to me soon or via email. But the idea is the same in that if you look here, you're plotting out the data and essentially you're gonna have them based on the increments. You're gonna do this process on the same graph. So like, for example, let's say this is at 30 degrees. I mean, you're gonna have increments. Oh, it's small to see. It's a bit small, but the idea is that you're gonna be able to see a trajectory. And it may be something like this as an example. It may even go down for some of you based on the lab recordings. But the idea is that you're plotting this out for all techniques, microplate reader, viable counts at 30 and 37 degrees. There should be the results already available. I have them already shown for you for our group for the microplate reader at 37 degrees. Practical 7, we'll wait to see if they share theirs soon. But in the meantime, you guys could get started with this to start plotting them out. Now, to answer the question as to what you do next, because you're going to get these all plotted out as a line or a curve, and essentially, you have two options as shown there. One option is that you use the slope. Essentially, you're going to make a slope of your curve. Let's say we're just focusing on our microplate reader results for 37 degrees. So if this is your slope, and you've plotted out all the lab averages as I showed through the guide, let's say these were your results, and then you made a curve, and you have a slope now in the middle, somewhere in between like this. You're going to use that slope, calculating using the formula there, which is log y2 minus log y1 divided by x2 minus x1. It's very simple in that the only difference from what you would have done like in calculus in grade 11 is that essentially you're adding in the logs, the logarithms, meaning that let's say this is y1. I'll just demonstrate it. Let's say you have y1 here and y2 here. x1 is going to go down here. x2 is going to go down here. Now, you're going to use those results of those essentially those time points or those dots that are in between time points greater than 15 minutes, right? And so the idea is that within that slope that you've already drawn or annotated on each curve, you're going to take the readings of log 2, so you're going to use the logarithm from your scientific calculators, minus log 1, and then divided by the distance between x2 and x1. If, for example, if your absorbances are essentially double, let's say it goes from 0.12 to 0.24, then what you do is essentially look and interpolate the location of x2 versus x1. So let's say this is 0.26 and this is 0.13. Then using x1, this is x1 and this is x2. The distance between these two, only if the absorbances or viable counts are exactly twofold, that's going to be your generation time, the distance between or difference between the two. And so just bear in mind that you're going to be doing this, but also sample or essentially your calculations on the other side of your semi-log paper. And you're going to do that similar to what I've said before your viable. So I'm going to just do this one more time in case anyone did not see it from the other side. Folks, if you look at the guide that I sent out earlier, it's almost the exact same for the viable counts in that you had 3 different plates, right? For each time point, you're going to take those that are within 30 to 300 colonies and derive essentially what you see here, the results of your colonies or your cell densities per time point. So just bear that in mind. And when your group is done, you can send those data points to me, and then be able to essentially do the next steps for today's practical, which I just touched upon for absorbance. And for example, if I redid what I talked about earlier, it would be viable counts, and then you put the units, and bear that in mind as well for your title with the species name or your title. The time point would be here in minutes. Remember, this is also semi-log paper, so you just have to bear in mind the range will be larger, but you could use essentially the first half for absorption and the top half for viable count, because the range will go like from 10 to the -7, 10 to the power of 9. 10 to the power of 6 to 10 to the power of 9. So essentially, the idea is the same. You have increments that go up, are like essentially on semi-log. So they essentially vary. Let's say, for example, this is 10 to the power of 6. This is 10 to the power of 7. Provide the details accordingly. But let's say you plot them out, the mean lab values. I'll give you instructions if this changes, but the point is the same. You plot them out, you get a curve, you get that slope, calculate that slope, and one thing I didn't mention is based on that slope, you're going to divide to get the calculation. dt or generation time is equal to 0.3 divided by that slope. So that should suffice for essentially that approach, which I'm going to show here. Let's say you have a slope log. Look at that formula, or the formula on the slide. Log y2 minus log y1 divided by x2 minus x1. That's your slope. Divide that slope from 0.3 divided by the slope itself. You'll get the generation time. If your absorbances are twofold apart, i.e. 0.13 versus 0.26, or like actually the same either way for values. But the idea is that if they're 2 times apart, then as long as the range you are able to calculate and interpolate, you should be able to essentially get the distance between x1 and x2, and y1 is essentially half of y2 to be able to be plotted easily. And then thereafter you get the generation time. You'll provide your results with calculations on the other side of your semi-log paper, and then your discussion will include the generation time that you derived, what you would have expected. Before this, talking a bit about the summary of your objective of the lab, just to remind me and to make it clear for anyone else who to essentially look at your report or assignment, and then talk about limitations. Limitations that I've talked about from the past, limitations that you may have noticed even from today, maybe counting colonies, maybe you double counted, or you weren't too sure, or other extenuating factors that we've talked about, volumes, cross-contamination, errors in antiseptic techniques, etc. So bear those important points in mind. So to highlight that, you pretty much have 4 curves, including the data that I've provided, and you can look at the lab manual if there's any other questions. Any final questions? Actually, Anna had one. Yes. Okay.
Should there be 4 points on the graph then?
Yes.
Just 4, right?
Just 4.
0 Nepal times 30, the Nepal times 60, and the Nepal times.
That's correct. Yes.
Okay. For the first one, for the microplate one.
Just to also highlight, the units are different. Remember, even the exponents are different. Absorbance readings are like 0.01 to 1, right? For ourselves. But for viable counts, we're going like 10 to the power of 6 to 10 to the power of 9. So the range is going to be much larger. Just to highlight that, so bear that in mind when you're drawing out your curves. It'll look more expanded for the viable counts and more thinner for your absorbance readings. But that shouldn't be too much of a problem.
And then for the absorbance readings, you said we're using the classes labs, right? Like
Yes.
the lab, so all of ours should look the same.
Theoretically, yes. It depends on how long your increments are, because from the paper itself, there's a way to divide essentially. So it may be thinner or larger. Yes,
And now-
generally. That's correct. But I do want to highlight you're doing it for both 30 and 37 degrees. So remember, 4 curves. There's different ways to do it. You technically can do both on the same page, all 4 on the same page. And how I would recommend it is like this. Where I'm looking at right now, where the red marker is on, that could be your absorbance for both practicals at 30 degrees and 37 degrees. Just show me with a different color so I know with a legend, okay, this is ours and this is theirs. And then similarly for the viable counts, you can have it on the top. So you technically could say paper and have 1 instead. 30 is done from the other practical and we're doing 37. So the class, everyone should have the results for the viable count. Practical 7, they're gonna provide us their results on Quercus. 30 degrees. And folks, if for example, you don't see the results, this is a good question. Technically speaking, the results should be posted. And if you, for example, are wondering, okay, the viable counts, how am I going to get it at 30 degrees? You should see it on Quercus. Our guides, or at least the guides I provided, and including the outline from Dr. Brunt, the materials that you have are already available. I just want you to be able to complete and send me your plaque assay results, the colony, like the number of PFUs for each plate, and or your mean average for each group. And then on top of that, I need your generation time viable counts. So that way they could also be uploaded in due time. Any questions regarding that as well? Yes. It's the aggregate data. I also realized, Tana, you had another question. What was your other question? Okay, so yes, it's aggregated just to make sure it's pooled. So you guys will be able to essentially, obviously doing this, with the mindset that you're doing this independently, each increment at a time. With your results, just to highlight again, calculations need to be included on the other side. Make sure that they actually reflect what you see from your graphs, because if they're, let's say, 2 seconds for generation time, that may be a problem, or if it's 20 hours, right? So bear that as well in mind, yes. Yes, so you need to send to me as a group all of your data like the data I talked about, your plaque assay. Was there anything else you had in that question or no? Yes, so send it to me as soon as you can. You can send it to me as a photo. Send it to me with your group number so I could keep track. And then it could be shared when possible, when best appropriately. Any other questions? Yes, please. 1 per group is okay. But I need to make sure all 6 groups, please share me your data, because it's very important that it gets uploaded by Friday. So please share it with me as soon as you can. Very simple. It's probably in your lab notebook. Just send me a photo, 2 photos of the plaque assay results, your mean values, and as well as your results for the viable counts. I'll grab some soap if it's not available. With this, really appreciate the time. It is 12 o'clock. You guys can clean up your benches if they haven't been done so already. If you're incubating, great question. So for the exercise 2, folks, exercise 2. Folks, for clean up, please pay attention. You clean up with 70% ethanol, as you're aware. Return back all items, the vials for the antibiotic discs, into the bins, as I showed. The pipette boxes where they were, and the tips before with the glass Petri dishes. For the exercises, microplate readers on now. Now exercise 2, Alta. Don't leave, folks. I want to make sure everyone is clean. Everyone has cleaned up their benches accordingly. Have you cleaned up everything? Everything? You ready to go then? Okay, actually I do, but I see plates there. Is that your plates?
Yes.
Okay. Okay, go ahead. No problem. Just let me know, folks, before you leave, but briefly, and that's perfectly okay. If you guys are in a rush, I understand. I want to highlight though, Wescadine, place your glass slides there from exercise 4. Petri dishes, rinse off. You can add in, place the discs from the Stephanie's test into the autoclave. Any used tissues, toilet paper, anything that's been contaminated into the autoclave. Pipette tips should be placed back, those that are used, into the bins down there, apart from the ones that are being used. So return back everything that you have. That should be it. Exercise 4. Yes. So exercise 2, incubating on not inverted. Don't invert it. Keep it as it was on into the second incubator, top left side. Please make sure that you've done that. Everyone done? Are you guys done? We place it into the incubator. You know what to send to me? Plaque assay viable counts. Ready to go. Make sure you wash your hands. Hopefully. All the best.
Yes.
All of them are ready to go. Have you recorded chemical models? Yeah, because otherwise I'll be able to. If anyone has to return back to their autoclave plates, you will find them here labeled. But please bear in mind that if you want to keep them, if any member of your group was absent from today, it's the result. You could have them. FLR has descriptions, very detailed descriptions on, as I mentioned, species, the conditions, the title, and methods and results. Look at that caption itself.
Yeah.
Oh,
Not like. But I think I'm fine. We only have 2 that are 30 and 200. So I just back to 6 shots. Nothing else.
On the microplates going.
If you do that.
Yeah.
I just want to have an example if and so apart from that, put it at the bottom right of the second page. This could be written off. They simply or yeah, so doing the mind field like
Okay, yeah, I just want to make sure that we're not using it.
yeah, you could use the plant data for the first
Yeah.
9.
Call.
Yes.
Okay, thank you.
If you can put pictures, please do so.
For this, are we, why did you actually?
Judges, please.
Oh, yeah, sorry, just, also technically. I don't want,
No problem, please here. Blue staff, please, on the line of guard, please, flow. All of that.
It's.
Yes. Sorry, first,
It's like 70 people. Yeah, I mean, I have so many. So many. I can correct. I agree.
it's okay.
I make it this course. I say it's a real.
If you get all of the smoke. All of them, well, yes.
So many. Cross blank for every single one.
I thought she made a question.
Yeah, or.
So they can start using.
So as we calculate the chemical that I just said, we have a lot of absorbance values for each time.
Then wait, can you take both?
Okay.
Yeah.
Like, so just time one, and 2, 3, 4, 5, 6, 9, so the rest we don't have to care about. Okay, up here. Okay, so once we get all of these laboratory values, we're putting them all into a graph on the central on paper. At times, they're at degree 30. 37, so for time one, and then where's 30? No, so we do 2 charts, 30 and 37.
So that's so they will have the same thing.
This is gonna have way more dots.
Well, that's because we're separating a bunch of you can have another chip from there. Should be a lot.
But if you get a lot of sort of value for each of these for 9 times, then we're having what, like, I don't know, it's like one dots.
What they're doing, probably should get right. Okay. I don't get it.
One time.
This one we're correcting here.
The number 6, so number 5, we probably, number 6 is
Let me show you.
Number 6 is the corrected data, and then number 7 is your lab average for each. Let me show. I want to. Let me.
Because I already did now we do the
okay, same thing as okay, so that's the graph, that's the variable cost. So I'm plotting the 4 points, my time at 0, time at 3. Time and final here, so it gets these 4 points, and then it's the same for the other groups. Okay, so that's what the left side of on the underside along the discuss everything we did. You want to on the back side of setting on section. Yeah.
Your aggregate.
So then
I just wanted to really, I'm just working to my life in such a I guess, check your like, how do you hit, essentially, for your, then behaving by that would be very violent.
So you could use it to essentially, yes. So do that for each of the, each of the Bible, or each of the,
one and two, or one and three.
1 to 4, all pretty much. You know, I know it sounds daunting, but believe it or not, this is easier. Last year was 6 groups. Yeah. Row 7 is essentially the lab. So think about
So here.
Yeah.
Three and four is so both of them. Adding those together, divide the square root of 7 times 5. And number 6 will basically be 5 and subtracting 1. That would be number 6. 7 is you're keeping all of your 6s and dividing them by 6 to get the final. So 7 is 12. Yeah. So 1, 2,
No,
3,
I thought it was yours and you just left it there.
Oh no, I actually thought. Barb, is this yours? I remember someone left it here. Is this yours?
Absolutely.
Okay, let's leave this.
I think just explain all of it. Would you be able to send like an announcement about what you expect by Friday? Or I can just write it down. So to summarize, you want
Bible count?
so 1 viable counts. So I did the counts. You just want. So this is fine when I say. Yeah, 10 to 5, 10 to 6. 10 to 7. This is perfect. This is what you want. That's 1. And then 2 was the Plaque Assay counts.
Take care, Garvin. Have a great weekend.
And second is Plaque Assay. And the rest we can do on our own time, like at our own discretion. But you need this for the. I'll try to get it done by tomorrow.
He has either of them can do it as well. Like any member can put the green rush. But sooner, but
Okay. I'm just like, sorry.
if you can remind me, I know you mentioned it, but in case like for you,
Yes.
but if you can just remind me so that way I can keep track. Or
I'll say like on behalf of 3.
yeah.
X and Y, this is Bible, this is Plaque.
And in case they want to also be kept in the loop, you can CC them. Yeah. If you don't,
Yeah, we made a WhatsApp chat. So kind of a.
perfect.
And then for the FLR, are you in a crunch? I wanted to show you my figure caption and then get your thing it on.
I can show it to you as I want. Is that okay? Where are you
Yes.
going?
Sorry, where are you planning to go? I have a tutorial at 2 p.m., but I'm on.
Let me just close some things.
I have most of it, like 3 things that I was.
You can show it to me actually. I just do you have it on your phone or?
I have it like. Oh, I have it up already.
Okay, okay. Let me in the meantime start cleaning up.
Yeah, of course.
It did. It's basically, I feel like the assignments in this course are really just like not explained well on the document. It's not by you, but in general, like I feel like we're kind of just left the bench for ourselves to figure it out. So I need someone to like sit there and explain it to me. What you did now, it helps me up. Just to formalize what I'm also government.
Yeah, no, I agree. Like it's, I think what she writes.
It's very vague.
Yes.
Yeah.
I'll try to see if like
I think the biggest issue with the FLR was the word count, like the page limits. Because I was trying to include like limitations, feature
Yeah,
applications,
yeah,
etc. But I was like, it was getting stuff.
I know. You know what? Do your best. I'll try to be lenient with
Like I go into limitations and stuff, but I obviously can't go Plaque Assay differs X and Y and then give an example. I just kind of like glaze over.
I would say, like, generally speaking, if you're able to highlight, let's say, 2 or 3 limitations and being able to, for example, say, okay, this is my limitation, aseptic technique, what could have happened, and how it could have been improved, that should be enough for me. 3 times, 2 to 3 times, depending on the level of detail that you're able to provide. Because certain limitations, you know from yourself that, and anyone else from the lab may know, okay, this is exactly what I did wrong, and how this could have messed or influenced my results. And then the next step is, okay, what could I have done better? And then just describing that with some extensions. Yeah. No. You can, yes. That's correct. If you can though have like one primary article as well in the intro, that would be nice. It gives. Okay, that's good. That's good. All the best. So I want to make sure you guys lose 10%, but I wouldn't. Like, I would pass it on not trying to do it. Try to email Dr. Brett if you can. I don't want to like because I really I do want to accommodate, but technically the question is like Dr. Mark may ask, oh, did come and give you access to the material for you? And then yeah, technically maybe like an issue that's fair then that you know considering the circumstances, but I would say do your best with what I provided, even if it hypothetically means that like you start doing it right now. At least you should be able to that FLR in about 5 hours time, because there's a lot of material that's visible. It can be
Between 3 and 5, yeah. Like if you sit there for about
and I could have a skim through if I have time. I'm a bit busy, but if you guys have problems or concerns, I'll try to at least compensate if I miss out on anything that you've asked for. I used to, but now I'm a bit busy with a lot of other articles, which is why it's a bit difficult. Let's see. Email as soon as you can if you have questions. But I don't want to be hesitant, but if you can, the sooner the better. No problem. Take care. That's okay. No problem. No problem. Have an amazing weekend.
Yes. I actually said for it to be the third step. Clear up everything. I want to make sure there's nothing missing.